you. My name is Sunitha Malapati, and I'm the Vice President of the CACNA1A Foundation. I'm thrilled to be moderating this session titled Charting the Course, CACNA1A Roadmap to a Cure. As we heard in the earlier sessions today, CACNA1A related disorders are very complicated given the size of the gene and the variability in the community's phenotypes. The path to treatment can feel daunting, but luckily the CACNA1A Foundation is dedicated to finding a cure for this rare disease, and we are supported by an extraordinarily smart and engaged scientific and medical advisory board. One of those members is Dr. Jeff Noebels, whom we are honored to have speaking with us today. I'm going to truncate his bio for the sake of time, but you can see his full bio on our website. Dr. Noebels is the Cullen Chair in Neurogenetics and Professor of Neurology, Neuroscience, and Molecular and Human Genetics at Baylor College of Medicine in Houston, Texas. He's also the founding director of the Bluebird Circle Developmental Neurogenetics Laboratory. Dr. Noebels received his PhD in neuroscience at Stanford University, his medical degree from Yale, and did his residency training at Mass General Hospital. I told you, we have an extraordinarily smart researchers on our side. His goal is to make a meaningful difference in the treatment of CACNA1A related disorders. And so we're putting him to the test today to discuss the essential research questions that need to be answered in order to guide us towards meaningful outcomes in patient-centric clinical trial design. You can go ahead, Dr. Noebels.
Thank you, Sunita. I'm happy to be here. I'm Jeff Noebels, and I've been thinking about this day for a long time. It's a very meaningful one for me and for the foundation, and I want to thank Sunita and Lisa and everyone who's working with the foundation for making this historic meeting come about. And so I'm looking forward to it. Now, they've assigned me a kind of a daunting task, which is to talk about the roadmap to a cure, but I've thought about this a little bit, and I think even though we're all coming from different backgrounds and looking at the problem through a prism at different angles, I think we can all understand and agree on some basic steps, but I want to make sure everyone is on the same page. So I'm going to talk about this map, which is not really a map, it's sort of a strategy of going from the gene to the target and the target to the cure. That's kind of obvious, but here's what's encouraging to me is, first of all, we're already on first base. We have our gene, and that's why we're all here together. And so we now have to go the rest of the way, and it's a journey of multiple steps. But despite that, there's a lot of work to do, but we know exactly what we need to do. So we know there are many variants in this gene. You've heard about them and how complicated they can get. That also means perhaps that we need to identify many solutions. It also means that, as you'll hear, that there are many solutions available, I think. What's encouraging to me, and I want everyone to understand, is this is not a linear process. A lot of people are working on different aspects of this at the same time. And so as we combine and as we share our information, we'll find out we're actually ahead of where we thought we were. The other thing about science, and many of you know this already, is that it's serendipitous. All of a sudden, maybe tomorrow, maybe next month, somebody will find something that really changes the way we think about it, and perhaps will even change our directions. And so for that, I think we all need to be flexible enough to take advantage of discarding ideas that don't work and jumping onto new ones that sound like they're promising. So this is a well-worn path, and I think we're all happy to be on it at this point. The other point, and I'm sure you've been saying this all day, is the exciting thing about this is the fact that we get to work together. And the amazing success of the Waze app is that they populate it with people who all share information with each other, and gets you down to where you need to go more quickly and accurately and avoid the dead ends and detours. So what we're all doing together is recruiting more people to understanding what our problem is and helping us work on it. In science, that sometimes takes some doing because people are working very hard on their own problem. But the more they hear about small successes and large ones, the more they will come and share their point of view with us. So we look forward to that. The other thing that we all know about is, we're wondering how long it's going to take to get there. And I'm sure the families are the ones who are most eager to learn that answer, and what their expectations are and what they're realistic about this research. And, I'm an optimistic person, but I think we have to rely on all of us together and brought together by the foundation to fill that glass to the top. So what we need to do is to look at the horizon, take the long view, set our milestones, and get to work. So here's the encouraging part, and you probably all are part of this, is that we have a solid infrastructure plan in place. And so these are all the steps, and more base hits and singles and doubles will help us build our database full of patients, perform the natural history so we have a better idea of what's going on disease-wise, to build the biobank that the scientists can avail themselves of and collaborate, and finally the translational research. And that's the part I'll be talking about next. But what's not on this slide, and I suspect you've been talking about it, but probably deserves to be on this slide, is what we also need is to build a public CACNA1A variant database. So this is actually drawn from the example of the Dravet syndrome sodium channel database, but they have designed a beautiful website where they accumulate all the information that we're hoping to get in a very nicely organized way. So I think if we can borrow this design and look and be able to set up a place where scientists and clinicians can go to understand and populate the database with more information. And as we go forward, we all know where to go to get it. This will allow us at a certain point, maybe soon, to sort of organize subtypes of the disease, and ultimately to allow people to quickly classify which patients are going to benefit from which therapies. So this will help us a lot. And if we haven't gotten started with this, then I'm happy to help. Now I'm going to take back sort of a little bit of history here, and it's appropriately in black and white because it was a long time ago, and explain to you why I'm excited about this particular gene. And that is, when I was studying epilepsy as a postdoctoral fellow, I needed to work on a problem that was real. And we had been studying epilepsy using convulsant drugs, which is very convenient, but is not why people have epilepsy and diseases. So I went in search of a spontaneous mutant model that had real seizures. And so I began to screen, actually for the first time, learn how to implant mice for EEG recordings, and then to start to screen a small library of neurological mutant mice. And the very first one I picked, there's the serendipity, turned out to be the tottering mouse. And what I saw was this amazing seizure discharges that had a spike wave appearance, and every time that happened, the animal would stop moving, and then would start again, just like childhood absence epilepsy. And the mouse responded very nicely to ethosuximide, which is a drug that works well for at least half of children with absence epilepsy. And I realized, and it's kind of amazing to think about it now, but we published this in the journal Science because it was the first time that epilepsy had been linked to a specific genetic locus. We didn't know what the gene was, but it was a Mendelian inheritance. And that had been very obscure. People at the time thought epilepsy was a multifactorial disease. The genetics were completely unknown. And you can see what happened as a result of that now in search of rare epilepsies, but turns out we can find a lot of single gene epilepsies. But it was the first time we could actually assign a clinical syndrome to the mutation in a single gene locus. So fast forward about 17 years, and that locus was cloned, and it turned out to be the PQ type channel that was mutated, a single base pair in that mouse. And then a few years later, it was revealed that it was a loss of function mutation that depressed the amount of calcium current entering into the cells. So what that does is set the stage for the generic question of how we go forward. And that means we look for different mutations in these genes, perhaps express them in mouse models, and then try to understand what comes after you mutate the gene. So the disease is more than just knowing what the gene is in the EEG. There's a whole biology that goes in between there. And as you can see in the bottom lower trace, that this is a biology that spans many orders of magnitude, starting from just a single amino base change in a nucleic acid, and then to the protein, and then trying to understand what that mutant protein does to a cell, trying to understand what the mutant cell does to the circuits, and finally, what does the mutant circuits do to the brain? Why does it cause that disease? And so you start by asking some basic questions. First, in this first part, what's wrong with the channel? And for that, we've always had, cells that we could study where we express the mutant into the cell and see what it does to the calcium current. Now we are very fortunate we can actually derive real neurons from actual patients with the disorder and look at their own personal mutation and see what it does to actual excitatory and inhibitory cells. And then the next step is the harder part, which is to try to understand now, what does that do to the brain? Why does it cause the disease that we see and not some other disease? And for that, we're lucky to have our friend, the mouse, which gets us all the way there because we can now genetically engineer these mice, follow them during development, and understand the disease. And it's complicated because what starts out as one single base pair error can alter the channel, but what the mutant channel does is create many different changes within the cell, and those mutant cells create many different changes within the circuit and the brain, et cetera. So there's really a complex natural history in the biology of the brain. We've talked about the natural history of the disease in the patient, and what's causing that natural history is this sequence of changes going on as the brain develops. And we need to actually understand that in order to fully rescue the error. The encouraging thing, though, even despite the complexities, that the target that we want to treat could be anywhere along this path. And I'll give you some examples. The other exciting thing, and why my glass is half full, is that we now know we have very powerful tools to think about this project and how to go forward. So starting from detecting that single amino acid change, we now have the technology to quickly get that done. When it comes to understanding what that does to the current in the cell, we have powerful technology, the patch clamping and using patient iPSCs to get a really accurate answer of what the mutation has done to the cell. In terms of drugs, we have a very sophisticated computer simulation of structure of these proteins and whether the mutation has altered the way it folds, and so does it reveal or close up a pocket where a drug could fit and work. And so we can learn a lot about that even before touching a drug or a mouse, and then using high throughput screening to find compounds that will actually match that desired effect. But then, of course, on the other hand, and I hate to say this, but there are many known unknowns. There are a lot of things that we know now we're not going to understand fully, starting with the idea, you've heard about these, of the variants of unknown significance. So you look at the base pair change and you can't figure out, based on all the models, whether it does anything or not. And you might measure something and you don't see a difference in the function of the channel. And so, but that doesn't necessarily mean it doesn't have one, you just haven't detected it. As well as there are other variants and other relevant modifier genes that could completely alter the way you interpret the genotype-phenotype relationship. So, it's fine to understand why a mutation in a certain part of the channel causes headache versus seizures versus ataxia, but many of the patients with that won't have that. And so these are the resilient cases that actually can really confuse us and slow us down in trying to understand. Nothing to do about that except to keep an open mind, look for it. We also know now that having an abnormal calcium channel can really change the way the brain develops. The circuits will develop differently. And it's important at what time this occurs, because there are these developmental critical periods in brain when, if the information is not accurate, the wiring will forever be abnormal. And so we need to take that into account as we think about the targets and the timing of the delivery of the drug. Finally, in the drug itself, you know, if everything goes well, we could design exactly a chemical that would fix the problem, but there are some channel configurations that might not be rescuable, including ones when it's deleted and there's no channel there at all. Of course, now we're fortunate because most of our disease is dominant, which means you still have a healthy copy of this channel, and there are ways of perhaps stimulating the generation of more healthy copies of the channel. But in fact, sometimes we may not be able to target the mutant channel itself. And then the last step is the one we know about, which is that not everybody looks the same. And we need to, although we've identified your variant, we need to know what is your disorder and will that drug that we designed fix all of the aspects that have gone wrong in this patient. Very important. So we really need to understand, and it's hard to acknowledge, but for every variant, how does that variant cause the disease? Now, we're not going to study 1,000 of them, but we may get some very general principles by looking at loss and gains of function, have them fall into groups. And that will really simplify the job, both for scientifically and clinically in terms of treating. So a little more on that subject. So these calcium channel variants contribute to a spectrum of comorbidity. So many people taking care of these children wish that epilepsy was the only problem. In fact, there are a number of other issues that come up, all as a result of a mutation in the PQ channel, but different patients have different presentations, and maybe they come in different order, in different sequence. But no matter what, these different variants, the reason they're doing that is they're affecting different networks of cells in a different way in different patients. So they're not always doing the exact same thing to the network we're trying to understand. And so that is something that we know, and we think we know why, because a lot of these channels are spliced differently in different cells, and there are other reasons too. But what it means is the challenge that we have, and that means, can we detect and rebalance excitability in a defined microcircuit without altering the others? So in a perfect world, we would give a drug and it would just fix the neurons that are not working properly. But actually, you might give the drug and it's going to affect all the circuits, but in a certain way. But in other words, can we learn to actually be so smart to create drugs with no side effects? And that's something that we don't know yet. But we'll try. That's the whole point. Now, another thing, and this is diving a little deeper for some of you, but PQ calcium channels are a little different than sodium channels in that they have a dual role. They not only regulate the excitability of the cells so that they will fire more frequently or less frequently, they're more or less excitable. That's key for very precise synchronization of neural circuits for cognitive disorders. It's all happening in the dendrites. But we also know that the PQ channel plays a very important role in the release of neurotransmitter at the completely opposite end of this neuron. Also tricky because it regulates the timing and the how loud a voice the neuron speaks to the next neuron and how quickly it can follow the instructions. So when those two things are wrong, then you're going to have a real problem in your circuit. The fortunate thing is that as we look at this diagram of the channel protein itself, we've long known actually that there are certain domains, certain regions where if we detect a variant in those domains, we know more or less where to look for the actual physiological problem. And so that'll help us going forward to continue to map these variants onto the protein itself. And then with basic scientists, understand what happened, how does that channel misbehave when it has problems in that particular part of the channel. Now, this is a slide for everybody because it really summarizes a major point, and that is that the PQ calcium channel, it's not like a light switch that turns on or off when you depolarize it. In this sort of amusing baby birthday card I found, there's your healthy calcium channel, PQ channel with an open pore where the calcium ions go in, and he's sitting in between these lipid bilayer in the membrane of a neuron. And a healthy one, he's happy and he does exactly what he's told with his other subunits that he plays with. But these variants, what they do, they don't open or shut the mouth. They cause it to go into very sometimes subtle different moods and different behaviors, and maybe sticking his tongue out, but sometimes these are subtle problems, and you can measure them, but they may not seem that impressive. But the fact is, if you spread this out among the channels that throughout an entire brain network, even a small change can actually end up having a pretty major effect on the behavior of signaling in that brain network. And so whether it interacts with the amount of current, how long the current lasts, or its ability to interact with other proteins, we need to understand this, that it's basically these mutations are more like dimmer switches rather than an on-off kind of thing. And that, of course, means that it's good because it means the drug can have a small, subtle effect, and it might make a big difference. So we still have much to learn, I think, but we know a lot more about the channel, I think, than the disease. So a lot of us need to focus on the mouse models to learn the second part of that journey, which is what do these mutant channels do to the brain? Now, this is a slide talking about what we do know from some of the preclinical drug trials that have taken place over the years, starting with ethosuximide, which I used and found that it was really effective in the loss of function tottering mouse model. It didn't do a whole lot to the ataxia, however, and it didn't do too much to the dystonia, the episodic dystonic episodes that these mice have. So it's sort of a partial response. And then other studies over the years, they didn't take the time because it's expensive and you have to have a lot of animals to look at all of the presentation of the mouse. But different drugs would have some effects on one aspect of the disorder, like the seizures, but either they didn't actually record the effects on ataxia, the dystonia, or it didn't do much. There was one study, which was mine, which early on I found to be remarkably effective, and that was because actually I didn't know what the gene was, and so we looked at the brain to find out neurochemically what was different about it, and we found that it had too many noradrenergic terminals, which is interesting. And we knew that there was a drug, 6-hydroxydopamine, that would reduce those terminals. And that actually, when administered on the first day after birth, gets into the brain and actually did a remarkable job of rescuing all of the phenotypes of this mouse. The only problem is that the window closes very rapidly, and we have no way other than direct injection of delivering this drug, which is not really a clinically translatable solution here. So, so we're still left with a bunch of partial studies here. And then there's a gain of function mouse model, which recently had a very elegant study using pregabalin, which is an epilepsy drug that targets the subunits, the regulatory subunits of the PQ channel. And they didn't report on the seizures or the ataxia, but it did reduce the cortical spreading depolarization episodes, which were the mechanism for the hemiplegia that happens with the gain of function mutation. But one thing I think you'll notice here is that none of these studies actually have targeted the PQ channel itself. So that remains to be done, and we're going to see that happen, I hope, in the future. So again, coming back to the idea that there are many downstream changes during development. So before we knew what the gene involved was in the tottering mouse, we looked at a lot of other neurochemical abnormalities, and different laboratories spotted a bunch of changes in the brain. And the question is whether these are actually targets that we need to worry about or not. So I call them what we call an endophenotype. That's something that comes after the gene, and it's something that the phenotype is inside the brain. And we don't know what it is. So they're EUs instead of VUs. And we don't need to yet to correct them, but we might need to. And so we'll have to figure out, it's a little tedious to understand what role these changes might have, but they could be important. And so we'll be looking at that in the mouse models that get studied from now on. These would be what we would call biomarkers for the disease, but we don't know if they're functionally contributing to disease or perhaps even trying to resist against it. So it takes a lot of time to sort that out. The one that we have paid a lot of attention to is the fact that mutating the PQ calcium channel actually alters the expression of another kind of calcium channel, the low threshold T-type calcium channels. And those have been implicated in actually causing the seizure. So we know that's a bad change. And we know that a PQ mutation that you're born with will reduce synaptic transmission. It will cause, in these two particular sets of cells in the thalamus, and inhibitory cells and excitatory cells, changes in their T-type currents. And those changes cause bursting behavior. And it's that bursting behavior, no matter how it's caused, that ends up triggering spike wave seizures. So when you're born with this mutation, it takes about 3 weeks for this to happen, and that's when the seizures start. And one thing you'll hear about in a nice talk tomorrow by CACNA1A, who's in our laboratory, is his work with a model that delayed the mutation. And so he was able to genetically engineer a mouse that would delete PQ channels in the young adults, so starting at postnatal days. So that would skip the early period of brain development. And the question, of course, is what would happen? And if you tune in tomorrow, you will find a beautiful answer to that question, but I'll give a little bit of the answer away, and that is that he got the full childhood absence phenotype, even though the PQ channel was reduced in the adults. And so there is no critical period, at least in terms of PQ, this particular model. So our future treatment strategies, and I apologize for the leaf blower outside. There are many different kinds of ways that we can think about and activating these targets. One is to either work on the PQ channel itself with activators or closers, most of which remain to be discovered for both CACNA1A and other channels that are close by. We can think about manipulating the nucleic acids with something called antisense oligonucleotides you may have heard about today already. These drugs actually last for quite a few months in the brain. They have to be injected directly into the brain. But in some diseases, this seems to be working, and you can actually modulate up and down the function of the channel that you're interested in. There are molecules inside of cells that actually are always modulating the behavior of PQ channels. And if we can find the drugs that will alter them or take their place, we can modify these channels from within. And again, I just want to say that sorting this out means that you know when to give the drug and for how long. So to summarize, ion channel mutations are not binary switches. They're infinitely flexible. Single variants create complex downstream trajectories and selectively remodeled circuits. The contribution of the mutant channel could change during development, but we know, at least in one model, that the adult PQ function is very important to not have the disease. So we have to rescue that somehow. Both the mutated gene itself and the circuit changes are targets. So it's not only, we're not limited. And what we need to do is build rich databases and multiple mouse models to connect each step from the defective channel structure to the patient. Now, I thought I'd just close on an inspirational note, and that is, perhaps many of you don't know what the first calcium channelopathy was. And it turns out to be in this creature, which is a single-celled paramecium. It's a unicellular organism studded with cilia that allow it to swim in the water. And about 100 years ago, a zoologist, Jennings, was looking at colonies of these mutants. And they normally swim forward until they bump into an object, and that bump triggers an inward calcium current, which allows the cilia to reverse and they swim backward. Well, he saw that in his mutant colonies, there was one set of mutants that could only swim forward. They would hit and they could not reverse. And so he named them pawn after the chess piece that only moves forward. And then it took about 75 years, but someone studied this and showed exactly that the pawn mutation lowered the inward calcium current compared to a wild type. And that was the reason that it couldn't back up. So the inspiring thing is that then a few years after that, someone looked at this again and found out that if you cultured these paramecium in a high potassium environment, you depolarize these mutants, that would induce a novel current that he nicknamed I queen. I is the symbol for current. So this is the queen current that restored the calcium current, you can see, when they were depolarized, and restored their backward swimming properties. And so he nicknamed, they're promoted to queens like you do in chess, just by depolarizing this. And another investigator showed that by promoting the depolarization promoted an auxiliary calcium channel binding protein, and that would help traffic more of the calcium channel subunit to cilia, and that's how it was rescued. So my last slide is simply to say, let's queen the tottering mouse and all the other models too, and see if we can, by following this and being optimistic and working hard, maybe we can get this guy moving again. So I'm going to end there and thank you all. And I hope there's time for some questions.
Thank you so much, Dr. Noebels. That was really, you've given us a lot to think about, especially as a foundation, and we'll definitely take you up on the offer to lead the charge on some of these potential solutions that you've outlined for us. So thank you. One question that we have in the chat is, you mentioned these critical time periods of brain development. What is the critical period to give a drug so that there is less damage that occurs? You know better than all of us that our kids often are diagnosed. It takes a long time for our kids to get diagnosed. So what are those critical periods when we're thinking about drug development and delivery?
Well, that's a great question, and also a difficult one to answer, because it could really depend, and that's always the nice answer you get, is that it all depends. So it depends on how much the brain is capable of reversing an early deficit. We know, and in fact, a Nobel Prize was given to Hubel Wiesel, who years ago showed that if you patch an eye beyond a certain point, it's very difficult once you remove the patch to regain normal vision in that eye. So the actual development of that visual pathway depended on normal input, normal activity. And they say that that firing begets wiring. So the two depend on each other. That's different in every part of the brain, but we think it's important. It's very important in some that may have to do with the spike wave epilepsy and probably the ataxia too. But a shorter answer would be that some of these variants are much more deleterious than others. Some of them actually hit the brain cells so hard that they either die or become deformed. There's some mouse models that show Purkinje cells in the cerebellum that are completely misformed. That doesn't mean they're irretrievable. Cells can do amazing things. They can reconnect. They can make new synapses and bypass the bad circuits. Some of you may know that if you take you can take an entire hemisphere of the brain of a child, and they can rewire so that they basically can use the opposite side almost as well, not quite as well. But so the brain is a plastic circuit that can recover from a lot of different kinds of injuries. So the answer is it really all depends on how injured the brain is. Obviously, in every case, we want to get in with a therapy as early as possible. But I just don't want people to give up and say, well, it's going to be too late. We'll never be able to do that. There's no reason to think that way. There's always a reason. As long as there's a brain circuit, there's a reason to think that you can enhance it or others to do its job. And so that's what keeps my hopes up.
Thank you. Another question in the chat: is the complexity of the gene a practical obstacle to finding a drug or a treatment? Do you believe that despite the diversity of the mutations or phenotypes, we've found enough patterns that a drug could help many different phenotypes?
I think it's likely. It's really hard to say because I can always imagine a case where that's not true. And I think we already see that cerebellar circuits that regulate your coordination and gait, they're different, and they have different chemistry. They have different wiring and neurotransmitters. They're different from the thalamus, that where we think a lot of the seizure generation occurs, and in the cortex. And so one drug may never, in some cases, remove every trace of the clinical deficit. But that doesn't mean there won't be a second drug for some people who are resistant to this first one. If you can gain, you just gain inch by inch, and if we can fix one part of it, we'll keep looking for the other drug that would fix the other part. And so I'm not at all pessimistic that patients can be helped in several different brain circuits at the same time. It's just a question of whether that one drug alone will do the trick.
Thank you. The next question is, how useful is it to repeat genetic testing given the rapid advances in genetics, especially if there may be multiple genes involved in some of our patients' disorders?
Yeah, so everyone's different, and they go to a different clinic because of the different clinical problem they present. And if they get genotyped, the evaluation stops at the first explanation for what that disorder was. So if you had hemiplegic migraine, they might be looking at the 3 genes that are known to cause that. And if you find one of them, you say, okay, solve that problem. But that doesn't mean that there aren't other variants in your genome, and there certainly are. And the question is how much attention is paid to them. Now, if you have a drug that will fix that person, they say, I don't care what else is the matter with me. And I'm not trying to predict the future. I just want to know that this genetic variant helped the physician to find the right drug for me, and we're fine. If you're resilient to those drugs, you have to dig deeper, and you have to say, look for the other suspects and say, this, first of all, the diagnosis may be inaccurate, it may be incomplete, more likely. And what else do we need to know about in you that could explain why you're resisting a drug? It's not clear to anyone yet why half of the patients with absence epilepsy don't respond that well to ethosuximide, whereas the other half are really great responders. So there are probably other genes in there that are sort of preventing you from getting where you want to go. And so it's a process of elimination, but we're only going to spend the effort on things that aren't working and try to solve them. So it's going to be a long time before we can make a total crystal ball evaluation from your entire genome and say exactly what you'll be resistant to, what you won't, what disease you'll end up having. I think we can explain the things that we see or try to. Prediction is a lot more difficult.
Thank you. And then the last question we'll take before we jump to the next panel is, you talked about this critical time period for our kids in terms of damage to the brain. How do you think that advocacy can change the timeline to genetic testing in a clinical setting? Do you have any ideas around making sure our kids get tested quicker and get their results quicker?
Oh boy, well, I think that depends on where you live these days. I mean, countries with national health care, I think have much more access to genotyping than those with private insurance, although I think in many cases that does the job here in the United States, but not for everybody. And also the question is, how quickly do you get to that point where you're genotyped? I think there's been encouraging progress at children's hospitals all over the country and the world know right away that the minute they see a seizure in a child, they should be thinking about genotyping. Why? Because they have access to the parents, and using that trio design of the affected patient, and they're comparing that patient to their parents, that trio design has been an extremely productive analysis, which has given us many, quote, de novo variants that have really pointed immediately to what the culprit gene is. When you don't have the parents, the older you get, the less likely they're going to be out in the waiting room or even available to get DNA tested. And so that already limits the number of cases you're going to be able to, quote, solve quickly anyway. And so the encouraging thing, though, is that people are starting to genotype older kids and adults in epilepsy clinics because there are gene panels and it's relatively straightforward to at least see if you have a known mutation in a known gene. So I expect that to accelerate. And I think the message just needs to get out. It's certainly out in the epilepsy community, but in other communities and movement disorders I'm not as familiar with. But the idea of looking for known genes in all of these disorders at the clinic, it's not that expensive anymore. And people are even saying someday it's going to be free. So I think we're going to see that. But all we can do is encourage everyone to seek those solutions.
Well, I just wanted to thank you so much for your time today and for your dedication to the CACNA1A Foundation. We are so grateful for what you're doing on behalf of all of our patient community. Thank you to all the participants today for joining and engaging with us. And we hope you'll join us at the next session, which you can access by going back to the main lobby.
Thank you.